So this is the story of how we discovered the modern theory of inheritance, and the guy who did it was a man named Gregor Mendel. Yes, and he was a monk in Austria. I'm pretty sure. Austria, very, very close to Czechoslovakia. It doesn't matter. The point was, his job was to tend the gardens. Monasteries often have a way of supporting themselves. They make brandy. They grow vegetables and sell them. The monks of New Skeet in upstate New York, they raise and train dogs. I mean, there's different... Yeah, right, monks of New Skeet, really cool. Not to be confused with the monks who say, neat! Sorry. Bad Monty Python joke.
I don't even know what that's referencing. So.
Good, you're a lucky man. Okay. But his job was to grow vegetables, and you know, farmers especially were very close to this idea of inheritance. They knew that, for example, if you wanted a big pig in your new litter of pigs, you're not going to mate the littlest pig from the previous litter with another little pig. You're going to mate the two biggest pigs that you have, and sometimes that would get you the biggest pig. Sometimes it wouldn't, and that's what we didn't really understand. We didn't know the connection, because we sort of said, okay, if you do this, it's going to work much of the time, but not all the time. And we really did think, you know, the blended, that was sort of the blended theory of inheritance again. That inheritance was like different colors of paint that you mix together. You know, you mix red and white together. You're not going to get red or white. You're going to get pink. Even though there were many, many cases where you would mix red and white together, you'd either get red or you'd get white. Sometimes you get pink, sometimes you get stripes. We really didn't know. And so what he did was he worked with plants, because he was in charge of the garden. And what he did was he kept very careful data, and he tried different experiments. And one of the things that he studied, that he's famous for, were pea plants. Those of you that know pea plants know that they have this interesting flower. I can actually tell you what, why don't I'll put a picture of a pea flower up there, because...
What year was this?
This was in the 1800s. The 1850s is my guess, but I'm not 100% sure. It's true. But they had a picture here of what Mendel could do with the flowers. But pea flowers, I'm not going to try to draw them, but they can either be fertilized by other flowers. We know how this works, right? The birds and the bees, you've got female flowers and male flowers. Female flowers have an egg inside of them, and male flowers have pollen, which is really sperm. And bees will rest on one flower, or other insects, and they will get the pollen on them, and they bring it to the other. And when the pollen lands on the, I'm going to say pistil, although it could be the stamen, I always get those mixed up. When they land on the pistil, do you know?
I am mixed up too, actually.
Yeah, of the female flower, the pollen bores into the plant and eventually fertilizes the egg. Now, pea plants can either be fertilized by other things, or they can also self-fertilize, because they have a unique situation where they have both. They have the male and the female reproductive part, the pistil and the stamen. They produce egg and pollen. And so, usually what happens is that they will be fertilized by something else, but if they don't, eventually what happens is the pollen is above the female reproductive part. I'm sorry, I don't remember what it is. But as time goes on, if it doesn't get fertilized, pollen will drop off of the male reproductive part and fertilize the egg. Self-fertilization. And so, what Mendel was able to do was he picked a trait. In this case, the one we're going to talk about is purple versus white flowers. And I'm just going to draw them like this, even though if you know pea plants, that looks nothing like a pea plant. But I wouldn't be able to do one correctly if I tried, and if I tried, it would take a lot more time. And so what he did was he found that if you mixed two different purple flowers, for example, and what I really mean is, let's say 200 purple flowers with 200 other purple flowers, that you would get a mixture of purple and we'll call these white, even though they both look white. Let's just, this really means solid purple, but again, I don't want to take all the time. Purple and white. Those of you at home, I hope you can see this. But what he found was he'd get a mixture, unless he self-fertilized them for many generations. And then he would get what was called a true breeding plant. And if you think about it, it makes sense. If you well, maybe it doesn't make sense, maybe it does. But what he would do is he would take one purple flower and mate it with itself. Sort of, instead of mating with another plant, he would take a brush and he would literally take the pollen, get it on the brush, and brush it on the pistil of the female plant, of the female part of the plant, and it would produce, after many generations of just taking the purple ones and mating with each other, he would eventually get to a point where it would be 100% purple. Or if he self-fertilized white flowers for many generations, he would get 100% white flowers.
So it's kind of like breeding for perfection.
Well, I mean, that's sort of, yeah, I mean, he was growing vegetables, he was growing peas, but he was also doing science. He wanted to be sure of what he had. So he noticed that the longer you self-fertilized plants, the more likely you were to get true breeding varieties. And so that's what he did. So then he focused on these. Once he got to this point, then he started breeding these together. And that's where the story gets interesting. Assuming that you think this story gets interesting, which I do. And so what he did was he took true breeding purple, here, I'll write it up here. True breeding parental cross, where he would take purple flowers, true breeding purple flowers, and true breeding white flowered plant, and he would cross them, and this was called the parental cross, or the pea cross, not to be confused with pea plants. That's just letter P, because it stands for parental.
So the true breeding parental cross is 100% purple.
Right. These were ones that were 100% purple, crossed with ones that were 100% true breeding. True breeding parental stock. And so, what did he get? Does anybody know? What was the next generation? Actually, don't say anything, because I'm just going to tell you. You can imagine what you think it was. What I would assume is that there'd be a mixture. It wasn't. It was purple all around. 100% purple flowers. And of course, he didn't just do one cross, and he didn't just get 4 flowers. He did 100 crosses, 1,000 crosses with 1,000 flowers each, and 100%. 100% purple. Now, what's that? Okay.
Was the dominant?
What?
Was the genes dominant?
Well, yeah, that's the, so looking back to what we know just from what we've learned yesterday. You should be confident in what you say. You were absolutely right. What we know now as residents of the 21st century, we know that that means that the purple flowers were dominant and that the white flowers were recessive, and that's absolutely true. But the thing is, he didn't know that. Nobody knew that in 1850. Nobody knew that. They had microscopes, but this wasn't figured out yet. And really, in a way, this is a testament to careful record keeping and observation, knowing it nagged at him that he knew there was a pattern, but he couldn't figure out what it was. So then, what he did was he crossed these because, well, okay, hold on. Let's get a little terminology going here. First of all, this was the parental generation. This is called the F1, or first filial, F-I-L-I-A-L. Right, it means son or daughter. Filial love is the love that a parent has for a son or daughter. And like, what's that?
Is that like filial piety?
I guess so, yeah. Yeah, right. Same thing. Or you even heard the term little filly, like a young horse. The filly, that's child horse. But anyway, okay. Well, what do you want it called? A little filly. It's but
Yeah.
let's move on, shall we? And so this is the F1 generation. So he did an F1 cross. And so, I'm going to erase these. Well, actually, I'll just erase two of them. So what he did was he crossed two F1 flowers. So, but let's talk here. Let's talk about how we represent dominant and recessive. Since we know that's where this is going, let's just get that going right now. Before, when we talked about this in terms of Legos, it would be, this would be the square Lego, and this would be the diagonal Lego, right? Isn't that what we did that in this class? Yes, on Monday. Monday? Yes.
Yeah, Monday.
Sure. Yeah. So the thing is, we don't use shapes in genetics, and what people would be tempted to do would be we use letter codes. And instead of having it be P for purple and W for white, which seems reasonable enough, but when you start talking about multiple traits that all might have different letters, it gets confusing. So what they do instead is they use capital letter and lowercase letter. So this would be dominant. You use the first letter of the trait, of the dominant trait. So dominant, in this case, capital P is purple. And the recessive is just the lowercase of the dominant one. And I know that's irritating. You want it to be like letter W, but it's not. Little p means, little p isn't here yet though, but little p would be white in this case. So the genotype, meaning the letters, the actual DNA that you have in there, the actual genes, the alleles, would both of, going back, I'll do this in red. Purple is big P, white is little p, little p. That's how we do this. We take a deep breath. I know, they look the same. Here, tell you what, I'll do it on a line. Big P, little p, little p. I guess that's the best I can do.
That works.
Okay. It's better when they're next to each other. So what we're crossing in this case, I'll do these in red. Okay. Nope. I'll do these in red. Big P, little p, crossed with big P, little p. Okay. So what did he get? Think back to, okay.
I have a question.
Yeah.
Can you have like if you have like two parents who are like having a kid and they both have recessive genes, can they are they both going to be different recessive genes, or are they going to be the same?
Yeah.
Because if they're different, then which one's passed on because they're both recessive?
Well, no, it's always in
How do you
relation.
have a more dominant recessive?
It's like tall and short.
Yeah.
You can have two short parents, but
Jack.
one's going to be shorter than the other. I don't know. When you have multiple alleles, there are other patterns of inheritance, like codominance, like purple and white make purple and white striped, or lavender. You know, but in this case, we're talking about simple dominance, which is different. So in this case, does anybody remember from what we did on Monday how this would work out? We did this. This would be the equivalent of square triangle cross with square triangle.
Yeah. It would be.
Shall we do the Punnett square?
Do it.
Do
Why the heck not?
Why the heck not? I can hear a bunch of people thinking a lot of reasons why the heck not, but that's okay. So what are we doing here? Punnett square, folks. You've got your two parents. So this would be paternal. This would be maternal. And so if they're both the same, one is big P, little p, and the other one is big P, little p. I guess I'll do it like that so you know what I'm talking about. And so remember, in a Punnett square, each square represents a different potential cross, because the pollen, the sperm that reaches the egg, is either going to be big P or little p, and so 50-50 chance. Maternal, same thing. The egg that receives the pollen will be either big P or little p. And so this box represents the cross between big P and big P. This box represents the cross between big P from mom, little p from dad, little from mom, big from dad, little from mom, little from dad. Each box is a cross of the ones that are above and to the side of it. And so what do we have here? It looks like we've got, for genotypes, 25% with the homozygous dominant, 25% with homozygous recessive, and then both of these 50% heterozygous. Notice how it doesn't matter if the big P or the little p is first or second. They're both heterozygous, which means they're both dominant. So it turns out that, to wicked oversimplify down to 4 young, %, white. He writes with the purple marker. Nice. 25% white, 75% purple. And don't forget, it wasn't just like when you flip a coin 100 times, you're not going to get exactly 50% heads, 50% tails.
Yeah.
But the more you flip it, the closer you'll approximate that. So for Mendel, I know we're getting kind of busy here, so I'm gonna stop in just a second here. Busy board-wise. So it was more like he would have, you know, out of 1,600, for example, 1,600 young, it would be more like, you know, 375 white and 1,225 purple, or, you know, 415 white and, oh gosh, 1,185 purple. Wow, that took longer than it should have. So, again, important things, and we're gonna shift gears here in just a second and shut this off. But first of all, there's the Punnett square skill that we're going to get a chance to work on. Luckily, I'm sure you were worried about that. But parental generation, F1 generation. By the way, what do you think this generation is called? F2, that's right.
And it just keeps going.
It would, yeah, right. If you did a third and a fourth, yeah,
Going again.
exactly. So this would be the F2 generation, the second filial generation. And by the way, F2 can come from either mating two different F1s or self-fertilizing the same F1. It doesn't matter.
What is fertilizing F1 and F2?
And I don't know what the hell it is.
The way that he proved his theory to himself, actually, was doing what's called a test cross. And that's exactly what he did. He crossed
Something with a something.
He crossed an F1 recessive. No, excuse me. He crossed an F1 times an F2 recessive. What does that mean? He crossed, in reality, that means he crossed big P little p with little p little p. And he theorized that if he was correct, that he would get 50% recessive. So big P little p times little p little p. Big P little p, little p little p, big P little p, little p little p. And so that's a test cross. So if you're not sure, for example, if I had a purple flower and I wasn't sure whether it was homozygous or heterozygous, I'd cross it with a white flower. And if this was homozygous, big P, you would get 100% purple flowers. If it's heterozygous, you get, that's, okay.